Welcome to part 2 of our chapter 5 lecture. So we're going to finish our discussion on chemical reactions. We're going to talk about a few different groups of chemical reactions, and these are just a small subset of the really diverse range of different types of chemical reactions that exist in our beautiful universe. And so we're only going to focus on a few different types of chemical reactions, and the reason for this is these are ones that are um, very common in biological processes. So the first one we're going to discuss is combustion. And combustion involves a carbon-containing molecule reacting with oxygen, and that produces carbon dioxide and water. So all of our metabolic processes that involve the breakdown of carbon-containing molecules like sugar or fats or proteins are kind of a combustion process. Now our body uses a much more set of chemical reactions, a much more complex set of chemical reactions to harness energy from like sugars and fats than the reaction that occurs when you just burn something, when you just light something on fire and burn it. But the end products are the same. So combustion reactions are very similar as far as how much energy they produce, as similar to the way that our body would break down similar molecules. And the end products are going to be the same. So you're going to start with some carbon-containing compound. You're going to need oxygen for the reaction to occur. So both of those things are going to be on the reactant side, your oxygen and your carbon-containing compound. And then your product side is always going to contain CO2, carbon dioxide, and water, H2O. So that's the major takeaway for a test, is to be able to recognize what's a combustion reaction. You're always going to have some carbon-containing compound and oxygen on the reactant side and CO2 and water on the product side. If you see those things, you know it's a combustion reaction. We should be able to balance these. We learned how to balance combustion reactions in a previous chapter. So a couple of cool facts about combustion reactions. They are a type of decomposition reaction. So we're taking a larger molecule and we're breaking it down into many smaller molecules. You typically have like a very, very big hydrocarbon molecule like we have in gasoline. You can have up to octane, which is like C8H18, and that's going to break down into a whole bunch of molecules of CO2. So they're decomposition reactions. Um, the energy of the products is always going to be a lot lower than the energy of the reactants. So these reactions are very exothermic or exergonic. So these reactions are going to be considered to be irreversible. There's going to be such a large difference in energy from the reactants to the products. And these are, for all intents and purposes, uh, irreversible reactions. So here we've just got some simple examples of combustion reactions. And our first one is probably the simplest example of a combustion reaction. That's our burning of methane gas. Methane gas is natural gas. This is a gas that most people use to heat their homes and heat their hot water and things along those lines. So this is what we produce from fracking. We're typically getting this out of deep fracking wells in Ohio right now. It's one of our cheapest fossil fuels and one of our more widely used fossil fuels. Now, just like any other fossil fuel, and methane gas is a pretty clean burning gas, but it does produce CO2, which is a global warming gas. So eventually we need to curb our use of methane gas if we're going to keep our population where it is right now. But it is still, for the time being, one of our cleanest fossil fuels that we can use. So again, a combustion reaction, you've got your hydrocarbon or your carbon-containing compound, CH4, reacting with O2 to make CO2 and water. Our next one down is C16H32O2. That would be a fatty acid. So this would be like your body burning fat. Now, if your body's burning fat, it's different from if you just burn fat. If you just take a match and light fat on fire, fat's an oil, it will burn. And the body breaks down fats in a very more complex, multiple step set of reactions so that it can better harness the energy, so that we can store that energy from the breakdown of fat or the combustion of fat using oxygen in ATP, which is our energy storing or energy converting molecule in our body. So these are very simplified versions of a combustion reaction, but in a biological process, your products are still the same. You're going to make CO2 and water in the end. So again, we've got that CO2 and water going on. That's if you see O2, a carbon-containing compound on the reactant side, and that CO2 and water on the product side, combustion reaction. Boom. All right, next one would be sugar. So we're just burning sugar. Sugar does burn if you just take a match to it, especially powdered sugar. It can be very flammable. In fact, there was a company, I can't recall where, but that made powdered sugar, and they didn't have very good cleaning practices in their company, and there was an explosion in the processing plant because they had powderized powdered sugar, and the smaller the grain of the sugar, so if you have like crystalline sugar that's pretty big chunk crystals, that's not as flammable as if you have it really finely powdered. And the reason is there's a higher surface area, so that more oxygen can get at the fuel. So there was a spark somewhere where there was a lot of powdered sugar in the air, and it was all mixed out in the air, and the stuff just exploded, and it killed a lot of the factory workers there. The final one I have looks a little bit different, but it is still a combustion reaction. So we're reacting just carbon with oxygen to make CO2 in this case. And this would be an example of like a really pure carbon source. Some examples, coal, kind of. Coal's going to have some hydrogen. It's going to have some impurities, some sulfur in there too. But coal is mainly just carbon. There's a lot less hydrogen and oxygen than in like methane gas or gasoline or something along those lines, like diesel fuel. But graphite would be another carbon-containing compound that's mostly just carbon. Diamond would be very difficult to burn, but theoretically, if you got it hot enough, you could probably get it to go. So you'd make just CO2 in that process. So that would be one example of an exception to the rule where you need to have CO2 and water in the products. And that's because in this case, there was no hydrogen in the carbon source at the start of the equation. So alkanes are one of our most excellent fuel sources because they just give off a lot of energy when burned. And the reason for that is there's a lot of stored energy between the carbon to carbon bonds and the carbon to hydrogen bonds. Those bonds are higher energy bonds. They have a higher potential energy. The carbon to oxygen bond is a lower energy bond. So alkanes, of all the different hydrocarbons that we're going to study throughout the course of this class, our alkanes are just carbon and hydrogen bonds. And those are going to be our highest energy molecules. When we start to look at other biological molecules like sugars and proteins and even our fats are going to have some level of oxygen already incorporated into the carbon containing molecule from the start. So like a molecule of sugar has less starting energy than a molecule of fat. And the reason for that is there's more carbon to hydrogen bonds in fat. Pound for pound, the fat's going to have more energy than the sugar because it's just starting with these higher energy bonds. So we have to point out some of how we show chemical reactions as chemists before we can go on to talk about our next few types of chemical reactions. And I just want to point out some additions to how we show things above the reaction arrow here. So this arrow typically indicates the direction that the reaction is going. So we're going from left to right here. We'll notice that there are some things above and below this arrow. So sometimes above or below the arrow, chemists can add in conditions that we were running this reaction in. So you might add the solvent. So you could say that this was run in water. So they might have H2O there. So we dissolve the reactants in water and then it made this other thing. In this case, what they're saying is we had the reactant and we added H2 gas to this. So they probably had it in a flask, like the reactant is a liquid. So they probably just bubbled H2 gas into the liquid and they said we had also a platinum catalyst. And what it did was it added hydrogen. So we broke this double bond and we added hydrogens. If you look, each one of these carbons, here and here, only had one hydrogen on them. Now over here, they each have two hydrogens on them. This guy's got one hydrogen here, another here. This guy's got a hydrogen here and a hydrogen here. So we've added H2. This is an example of a hydrogenation reaction. In biochemical reactions, sometimes we'll see this curved arrow. And this curved arrow is really often used when we have an energy coupled reaction. So we'll use something like ATP. Sometimes there's something called NADH or NAD going to NADH. And so this is essentially stating that energy has been transferred during the course of this reaction. So ATP is one of the biggest energy transferring molecules in our body. Our body makes ATP from the breakdown of sugar. And interestingly, we have to spend a small amount of ATP in order to start to break down sugar in our bodies. You can kind of think of it as like the spark you need to get the fire going. So ATP is a high energy molecule. ADP is a lower energy molecule. So ATP is adenosine triphosphate. ADP is adenosine diphosphate. So if you look at the reactant here, this is glucose, and we've got an alcohol group down here at the bottom, and we've made what's called a phosphoester. So we've added a phosphorus to the oxygen from that alcohol. So this oxygen that's here is that same oxygen. It was right here, and we just stuck this phosphate group on there. So the adenosine triphosphate, it had 3 phosphate groups on it, has become adenosine diphosphate. The phosphate, the high energy phosphate, has been transferred to this glucose molecule. So this is now called glucose 6-phosphate. When we get into our sugars chapters, we're going to learn that that's because these are numbered. So this would be carbon 1, carbon 2, all the way down to this carbon here is carbon 6. So that's why this guy is glucose 6-phosphate. That's more information than you need to know for your test, but this is how we show when other molecules are involved in a biochemical reaction. We'll often show this curved arrow here and show what's going on. Then we'll also show the catalyst involved. So this is catalyzed by the enzyme hexokinase. So this is just a table that summarizes what we've already discussed. You don't really have to memorize this table for the test or anything along those lines, but it's kind of good for your own knowledge to kind of understand the different types of reactions and how we might label reaction equations depending on what type of chemistry we're talking about. If it's like a general chemistry reaction versus organic chemistry or biological chemistry, we might use slightly different writing cues in our chemical equations to discuss what's going on. Our next type of reactions that we're going to talk about are oxidation reduction reactions. These are extremely important biologically. So these reactions are involved in everything from metabolism of our proteins, our sugars, energy production in the body, to intracellular signaling. There's a lot of different redox reactions that go on in the body, and it's really important to be able to understand what is reducing, what is oxidizing. Also, a lot of the things that we use to sterilize equipment, medical equipment along those lines, we're going to use an oxidizing agent. Oxidizing agents break down biological molecules or oxidize biological molecules, including the lipid bilayer of cells and bacteria, proteins involved in those cells or bacteria, DNA. You can oxidize DNA and destroy the DNA. So single-celled organisms and viruses that might be on a surface will get hit harder by an oxidizing agent than like our skin would, because we have a thick layer of dead cells on the outside. You can oxidize your skin a little bit. It's not going to kill you. You know, the poison's in the dose, but for a single-celled organism, a small dose is a huge dose. So important to understand these oxidation reduction reactions as we go forward, because we're going to be seeing them again and again. There are 2 different types of oxidation reduction reactions. There are oxidation reduction reactions involving metals and oxidation reduction reactions involving nonmetals. So the first thing we're going to talk about is our metals chemistry. This is our inorganic chemistry. So when you have metals involved in your inorganic chemistry, this is going to when we talk about redox reaction, we're going to be following the movement of electrons. So an electron gets given or taken away from someone. That's what we call redox chemistry. We've already studied this when we studied how salts are formed and how ionic bonds are formed. But now we're just going to name the different players in this. So in organic chemistry, though, we're going to see that this is, instead of the movement of electrons, we're going to look at the movement of oxygen and hydrogen. So if you're adding oxygen versus, or taking away oxygen versus adding or taking away hydrogen, we'll have redox chemistry. All right, so the simplest way, if you have a metal, the guy who is oxidized gains or loses an electron. So sorry. And the guy who is reduced gains electrons. So we'll look at an easy way to remember this, and we'll show you some examples here. So again, atoms that lose electrons during a chemical reaction are said to undergo oxidation. So if we take a look up here, we've got iron. Iron in its elemental state is just iron solid. It's not bound to anybody else. So we know that if you're in your elemental state and you're a metal, your charge is a big 0. We have oxygen gas. We know that oxygen in its elemental state also has a charge of 0. We've got an equal number of protons and electrons in both of our reactants. Now we're making ionic bonds. So we're making a new ionic compound. And if you remember, when we want to figure out what the charges of these guys are, we have to trade these lower numbers over here. So our iron is going to have a charge of +3, and our oxygen is going to have a charge of -2. So we can do the math to remember this. If you remember, oxygen's in group 6 of the periodic table of the elements. All of our guys in group 6 have a charge of -2. It was something we learned in a previous chapter. And then we can do the math there. You've got 3 oxygens, so you have a total charge of -6. Therefore, if you have 2 irons, you'd have to have a +6 from those 2 irons. So the charge of each iron must be a +3. So now we know that somebody's gained electrons and somebody's lost electrons. So which was it here? What happened to iron? Iron started with a charge of 0. He now has a positive charge. If you lose an electron, that's a double negative, right? I just drew 2 minuses there. Electrons are negatively charged. You got this E -1, right? So if you lose an electron, it's minus a negative. You've become more positive. So who lost the electron? Iron lost electrons. He became more positively charged. Oxygen gained electrons. He became more negatively charged. So if iron lost electrons, and this says atoms that lose electrons during a chemical reaction are said to undergo oxidation, then iron was oxidized. So we would write oxidized under that iron there. Then oxygen has to have been reduced. So you're always going to have one guy that was oxidized and another guy that was reduced. You're not going to have both guys on the same side of the equation being oxidized at the same time. Somebody's got to get oxidized and somebody's got to get reduced. Those electrons have to go somewhere. They can't just disappear into thin air. So if one guy loses them, another guy has to gain them. The next part can get a little tricky or confusing for people. And that's which guy is the reducing agent versus the oxidizing agent. Now we said iron was oxidized. So who oxidized him? Who took his electrons? Well, oxygen took his electrons. So that's why we call him the oxidizing agent. So oxygen was reduced, but he is the oxidizing agent. So in order to get the oxidizing agent and reducing agent thing, all you have to do is figure out who got oxidized, who got reduced. So we're going to name on the product side, we're going to write iron was oxidized. I'm just going to write the ox thing there. And oxygen was reduced. Now we're going to flip that on the reactant side. If iron was oxidized, he was the reducing agent. Now I'm just going to write AG for agent here. You guys can figure that out. He was the reducing agent, and oxygen, since oxygen was reduced, he was the oxidizing agent. Oxygen oxidized iron. Iron reduced oxygen. And that's why we call them the reducing agent and the oxidizing agent. I really like this acronym. I'm going to show you another acronym in a second here, but this is the one that I learned, and it's Leo the lion says grr. So I just remember your Leo grr. Losing electrons is oxidation. Gaining electrons is reduction. So we've got two more examples down here. We've got sodium in its elemental state, so it's neutrally charged, goes to sodium plus. And we can see that that plus happened because we lost an electron. An electron is given off. So sodium has lost his electron. He's become oxidized. The next guy down, we've got chlorine. This is chlorine gas. He has two electrons bump into him, and you get two chloride ions out of this. So each chlorine atom has gained an electron. The chlorine has been reduced. So if we react sodium metal with chlorine gas, who is the oxidizing agent? What do we think? Take a second and try to figure that out. Pause it if you have to. Then I'll tell you the answer. The oxidizing agent is chlorine. Because remember, we have to look at the guy who was the opposite, right? He was reduced. So chlorine was reduced. He took the electron. That makes him the oxidizing agent. Sodium was oxidized. Sodium's reacting with chlorine. So if sodium is oxidized, who oxidized him? Obviously chlorine. The other guy he's reacting with. So you've got sodium would be our reducing agent, and chlorine would be our oxidizing agent. So a lot of these basic patterns you guys have already kind of learned in previous chapters in these inorganic oxidation and reduction reactions. Our metal atoms are going to be the guys that lose electrons. We all know that our metals take on positive charges. So our metals are going to get oxidized. So all of our metals, you guys on the left side of the periodic table, the elements, they're going to get oxidized. And what kind of agents does that make them? If you're getting oxidized, then you're the opposite kind of agent, right? So if you're getting oxidized, you must be a reducing agent. Our nonmetals, the things on the right side of the periodic table of the elements, they gain electrons, forming anions. So they get reduced. If they're getting reduced, that makes them the oxidizing agents. They're doing the oxidizing. So oxygen, chlorine, fluorine, bromine, iodine, these are all oxidizing agents. So a lot of the things that we use as oxidizing agents to sterilize materials or work surfaces or tools or things along those lines, especially in the medical profession, or sterilize your skin, we're going to use strong to weak oxidizing agents. So you don't want to use a super strong oxidizing agent on somebody's skin. So what kind of oxidizing agent do we rub on somebody? We'll use iodine. It's not such a strong oxidizing agent that it's going to be a poison for the person. So you wouldn't take and rub a whole bunch of bleach on that person's arm before you stick a needle in their arm. Bleach would be a strong oxidizing agent. It's hypochlorite. It's got chlorine and oxygen in it. So these things are going to attack biological molecules through redox reactions. And so we want to use a weak enough one that it's not going to hurt the person, but a strong enough one that it's going to produce the effect that we want to sterilize, kill the bacteria. So one way to look at oxidation and reduction reactions in biological systems is and how they work and why they're important is I compare them to like electrical lines outside that power our homes. In an electrical line, you've got some metal that is able to conduct electricity. So it's able to pick up an electron and then hand off an electron to the next metal atom in the chain. So that's how metals act as electrical conductors. They're able to pick up an electron and transfer it down there. We use copper often in our electrical lines as a really excellent electrical conductor. Iron's also an electrical conductor, and iron is used in a protein called cytochrome C or cytochrome C and cytochrome C oxidase that are involved in ATP production. And so cytochrome C and cytochrome C oxidase are involved in taking electrons, using those electrons to do work, just like an electrical motor would, or like your ceiling fan in your house. and they take these electrons. As the electron goes from one iron to the next, it gets transferred from an iron to an oxygen, and that makes a shift in the way that the protein is shaped, kind of like a machine might move or like a motor might move, and that drives protons, or H plus, through a membrane in your mitochondria. And these protons all build up on one side, and they get crowded, and then they rush back through this open channel that spins like a turbine, and that makes ATP. So that's part of our power generation. So if you think about oxidation reduction reactions, kind of think about these electrons move from one atom to the next, and there's power in that. Just like there's power in our electrical lines that we use to power our homes, to power our, the internal workings of our vehicles, our computers, our tablets, or your phone, or whatever it is that you're using. Oxidation and reduction reactions in organic molecules are still all about transfer of power, but it's a little more subtle. It's not just, you can't just look at how the electrons move. We're looking at whether you have oxygen or hydrogen bound to carbon. So when we're talking about carbon-containing molecules, remember we mentioned that if you have that carbon to hydrogen bond, that's a very high energy bond. But that carbon to oxygen bond, that's a lower energy bond. So in organic molecules, the more oxidized you are, the more oxygen you have bound to you, the lower energy you are. So in biological molecules, there are all of these enzymes that take carbon-containing molecules, and they can either oxidize them or reduce them. And this is our body's way, or nature's way, of storing potential energy in chemicals. So you can take a lower energy molecule like CO2, and plants then reduce that. So they're going to remove oxygen from the carbon. So the definition in organic chemistry of oxidation and reduction is if you've gained oxygen, you've been reduced. If you gain oxygen, you've been oxidized. If you lose oxygen, you've been reduced. You can also look at hydrogen. Now for hydrogen, it's the opposite. Typically, if you're gaining oxygen, you're losing hydrogen. So the full definition is an organic molecule is oxidized if it gains oxygen or loses hydrogen. And is reduced if it gains hydrogen or loses oxygen. So let's take a look at this example down below here. So we're going to start with an aldehyde group. That's a carbon with 2 bonds to oxygen. A bond to R would be some other carbon containing part of the molecule, and 1 hydrogen. So we've got 2 bonds to oxygen and 1 bond to hydrogen. So if we move in this direction, our reduction would mean that you lose bonds to oxygen. So whereas we had 2 bonds to oxygen to start with, we only have 1 bond to oxygen now. As you see, we only had 1 bond to hydrogen. Now we've got 2 bonds to hydrogen. So we've got 2 hydrogen bonds there. So that would be reduction. We've been reduced. This molecule is higher energy. There's more energy stored in this molecule than there was in this aldehyde. Now, oxidation, you remember we use oxygen to burn our food. To use it as fuel, we have to breathe in oxygen. So if you're adding oxygen, you're being oxidated. And these are going to be your lower energy molecules. So our aldehyde, in this case, got converted to a carboxylic acid. And this carbon has 1, 2, 3 bonds to oxygen now. And he lost a bond to hydrogen. This bond here was a bond to hydrogen. Now it's a bond to oxygen in this carboxylic acid. I'm not going to sit here and read through this entire slide, but this gives some examples of oxidation reduction reactions in biochemical pathways and how different things are processed. None of this will be on the test, but this just gives some additional information. So if you'd like to pause this and read through there, feel free to do that. So this is a very common example of an oxidation reduction in the organic oxidation reduction in biological molecules. So there's this molecule called NAD, or NAD plus, or NADH. And NAD stands for nicotinamide adenine dinucleotide. So it's got two nucleotide groups, and we'll talk about what those are when we get into our DNA chapter. And this guy is commonly used in oxidation reduction pathways. So if we take a look here, we're starting out with an alcohol. So this carbon has a bond to this carbon, two hydrogens, and this oxygen. I know we didn't do a whole lot of discussion on these condensed formulas, but that's how that works out. So we've got a carbon with one bond to oxygen, and over here we've got two bonds to oxygen. So this guy has been oxidized. So in this case, this alcohol has been oxidized. He gained bonds to oxygen. NAD was reduced in this process. He gained a bond to hydrogen. Remember, things are flip-flop between oxygen and hydrogen. So if you gain a bond to oxygen, you've been oxidized. Whereas if you gain a bond to hydrogen, you've been reduced. And it's the reverse if you go the other way. So if you say, if you lose a bond to oxygen, you've been reduced. If you lose a bond to hydrogen, you've been oxidized. So the next type of reaction I want to talk about is a condensation reaction. This happens when you're building biological polymers. So things that we use, the body uses that are polymers include DNA and proteins commonly. DNA, RNA, proteins are going to be the most common polymers. This same chemistry will be used to fuse fatty acids together with glycogen or other backbones, so that we have a you can form your lipid bilayer or different fatty acids that are used throughout the body. So in a condensation reaction, we're joining two molecules together and we produce water in the process. Now remember, our combustion reactions also had water as a product. So how do you tell the difference between a condensation reaction and combustion reaction? So you're going to look to see if you've got water. We've got water there, so you're talking we either have a condensation or a combustion reaction going on here. But we don't have CO2 also is one of our products. Remember, in a combustion reaction, we'll always have CO2. In a condensation, we don't. And the other thing that you'll notice about a condensation reaction is we started with two smaller molecules and we're fusing them together to make a bigger molecule and the water. So that's another thing to look for in a condensation reaction. So when you think of a condensation reaction, think about when you get condensation, like on your car in the morning, or water condenses on your grass to make dew in the morning. So think about water appearing out of nowhere, or seeming to appear out of nowhere, or seeming to appear out of thin air. You're producing water in a condensation reaction. So we get this water as one of our products. Now, hydrolysis is the exact opposite of condensation. It's just the reverse reaction of condensation. So in hydrolysis, your water is going to be on the reactant side of the equation. So you can see we have a double arrow here. So we've got more than one reaction going on. This is a reversible reaction is what we're saying here. So here we have adenosine triphosphate is one of our reactants. Water is another one of our reactants. And when it's going in this direction, this is hydrolysis. The water is getting split into two and is used to split this molecule apart. So you'll also notice we start out with this one big molecule, and now we've split this big molecule into two pieces. Sometimes this hydrogen here will be fused to one of the two pieces. Other times it will float off into solution, and this makes things a little bit more acidic. When you have this H+, this is like acid. So when you're burning a lot of ATP, you can generate a little bit of acid, and your body has buffers to buffer out that acid in the cell. The carboxylation reaction won't be on our test, so I don't expect you to memorize this guy, but the carboxylation reaction is important biochemically. It allows us to add a carboxyl group, and this particular procedure is used in several different metabolic pathways. So, for example, this is involved in converting pyruvate into oxaloacetate, which is one of the steps in the citric acid cycle. So this is involved in metabolism. Phosphorylation and dephosphorylation is another term that you'll hear when we're talking about biochemical reactions. And all I really want you to remember is that if you've been phosphorylated, you've had this phosphate group added to you. If you've been dephosphorylated, you've had this phosphate group removed from you. So this is happening. In this case, we're going in this direction, and there's an enzyme called alkaline phosphatase. So it's kind of got built into the word that it's adding phosphate somewhere or removing phosphate. And so which one is this here? We're going from left to right. Are we phosphorylation or dephosphorylation? This one would be dephosphorylation, because whatever this R group is, this could be like a whole big protein or something along those lines. It's having that phosphorus removed from it. This is used in intracellular signaling. It's sometimes used to turn enzymes on and off. So the body will add that phosphate group, or the cell will add that phosphate group, and that'll make a conformational shift. So the protein, when I say conformational shift, I mean the protein that's had the phosphate group added to it changes shape. And I'll try to send you, we'll send you guys some videos of that, or we'll talk about that in our in-class problem solving sessions too. So if we're going from left to right here, this would be dephosphorylation. And if we had the reverse reaction, so if we had like that double arrow thing going on, the other direction would be our phosphorylation. We're having the phosphate added to us. We talked a little bit about alkanes when we were doing our combustion reactions, and I just want to remind everybody what an alkene is. So if you remember, alkane was you had a hydrocarbon, so often our fatty acids are alkanes, and alkane is a hydrocarbon in which you have only single bonds between the carbons and of course the hydrogens. Hydrogens can only make one bond. But an alkene has double bonds between at least one pair of the carbons to be an alkene. So alkenes have some additional chemistry that they can undergo. When we have an alkene in a fatty acid chain, that's what we call an unsaturated fatty acid. So if we have a fatty acid, we usually have like a carboxylic acid group. So we'd have an oxygen double bound to carbon, and then you'd have another oxygen here, and this guy probably got a negative charge to him, or he's got a hydrogen. Then you got this big long chain hydrocarbon coming off. Well, if you have a double bond in there, that would be what we call an unsaturated fatty acid. If you didn't have the double bond, that would be a saturated fatty acid. When we say saturated, we mean saturated with hydrogens. Because if you have all single bonds, you can have more bonds to hydrogen. If you throw a double bond in there, that means those carbons are using one of their bonds to bond again with their neighboring carbon. So there will be fewer hydrogens that can be attached to that molecule. So when we say unsaturated fatty acid, we mean it's not saturated with hydrogens. And that's going to be important when we talk about additions to alkenes, because one of the things that people in the food sciences do, which is a little bit tricky, is they're going to add hydrogen to an unsaturated fat. They're going to say it's hydrogenated. So they'll trick you by saying that you've got some sort of vegetable oil. So they'll say hydrogenated vegetable oil. And what they've really done is taken a healthy unsaturated fat and turned it into a saturated fat. And they do this because it makes it taste more like butter. It makes it taste more like a fat from an animal, because most of our animal fats are saturated fats. We tend to store, as animals, we have to move our fat around. When we store fats, it's the saturated fats are higher energy per pound. So it makes more sense if you're going to be an animal and have to move that fat from place to place with you to carry something that has more energy per pound. Whereas if you're a plant, you're not really going anywhere. You don't have to worry about moving your energy storage. So it would be okay to store more of your energy in like the form of glucose or an unsaturated fat that's a little lower energy than a saturated fat. So these alkenes are double bonds, and you can have additions to these double bonds. So the body can take an unsaturated fat or a double bond and add hydrogen to it. So if you had like H2 in the place of this XY, we could add 2 hydrogens. So we would break one of these bonds in the double bond. That would give us room to make new bonds to this hydrogen. So this would be an addition to an alkene. And we'll talk about some specific types of additions to alkenes. One of those types is what I was just discussing, which is hydrogenation. So if you just think of the word hydrogen is in hydrogenation. So this would be if you are adding hydrogen to a molecule, or H2 to a molecule. Don't confuse this with hydration. Hydration would be like if you're adding water. So hydration sounds a whole lot like hydrogenation. But just remember, hydrogen is literally spelled in the word hydrogenation. So you're just adding hydrogen. If you're removing hydrogen, we would call that dehydrogenation. Now, if you have full hydrogenation of a fat or an unsaturated fat, you're just going to get a saturated fat, and that's fine. But if you get partial hydrogenation, you can get this kind of nasty product called a trans fat that our bodies have a hard time metabolizing, but they taste like a saturated fat. And for a long time, we didn't know that these were bad for you. We didn't know that our bodies couldn't break these down, or that they might build up in your body and cause heart disease. But we were making these things with like platinum, nickel, or palladium catalysts, and you add these platinum, nickel, or palladium catalysts, and you bubble in hydrogen to the mixture of your fat, and you get something that tastes more like butter. So they were taking these cheap vegetable oils, or these vegetable oils that were supposedly a lot better for you. They were a lot better for you because they're unsaturated. They don't stack up in your arteries as well, and they were turning them into things that are shaped more like a saturated fat. And we discussed in previous chapters when we were talking about molecular shape, the saturated fats are these long straight chain hydrocarbons, and they stack together really well. When you add a double bond, particularly a cis double bond to that molecule, you add a kink to the fat, and that's going to make it harder for the fats to stack on one another. And so these unsaturated fats are more healthy. When you go and hydrogenate them, you've kind of taken away that property of the fat. So I don't really like the way that the book chose to draw this molecule, because they didn't get the bond angles right on this top guy up here. And they did that so that it would fit in the figure a little more nicely, and so they'd have what looks like a straight molecule. But this is palmitoleic acid, and this is a cis unsaturated fatty acid. So you can take this guy and convert him into palmitic acid, which is just a saturated fatty acid. This is a little less healthy for you for the reason that these molecules are really straight and they can stack really well on top of each other. Now this molecule in reality looks more like this. I'm going to draw this circle here just to represent this here, because I don't want to draw the whole oxygen, blah, blah, blah. But you've got your long chain hydrocarbon here, and when you get to that double bond, it actually does something like this and makes a bend in that molecule. So it doesn't just keep going straight like is shown here. That's incorrect. This actually is going to have an angle here, and the next bond is going to come off, and it's going to go off in that direction. So it's kind of poorly drawn there, but this is why this doesn't stack up. These guys wouldn't stack up as well as a whole bunch of straight sticks. If you think about it, you have a whole bunch of straight sticks versus a whole bunch of crooked sticks, it's a lot easier to stack the straight sticks. So these build up in your arteries a lot better than these do. These guys tend to be, the saturated fats tend to be solids at lower temperatures, and these guys might be liquids. So this might build up a solid gunk in your arteries, and this wouldn't. Now the problem with a trans fat is it looks pretty much like the cis fat. You've got your double bond there, but this molecule now, because it's trans and the carbons are on opposite sides, I'm going to draw our hydrogens in here just to show you this. You've got your hydrogens on opposite sides of the double bond. Let me clear some of my stuff out here. And you've got in this guy, who is the cis, your hydrogens are going to be on the same side of the double bond. So that little bit of difference between cis and trans fat is huge. As you can see, if this is where the double bond is, I'm just drawing a line through the plane of the double bond. So in this case, both of my hydrogens are on the same side of the line. In this case, this hydrogen's on this side of the line, and this hydrogen's on this side of the line. So that's what makes it trans, which is opposite. This is make this cis, which is the same. So the cis fats are natural. If you're going to have an unsaturated fat that was made in nature, it's going to be a cis fat. These are synthetic, the trans fats, and our bodies don't have enzymes to digest these or break these down properly. We don't clear these from our body the same way that we clear a cis fat. This one might get a little bit confusing. This guy is called hydration. And remember, it's not the same as hydrogenation. And we also talked about another reaction that had water involved, which was condensation. And so condensation versus hydrolysis. In this case, hydration is the addition of a water molecule to a double bond. So in order for it to be hydration, specifically, you have to be adding a water molecule. So you have to add 2 hydrogens and an oxygen to the carbons involved in the double bond. So we're taking and breaking this double bond, and we're adding an OH and an H, which adds up to H2O. So that's hydration. So if we remember, if we want to talk about the differences between all the different reactions that we've talked about with water in them, let's break that down. In combustion, our products were H2O and CO2. Is CO2 involved in this one? No. So this is not combustion, this is different. In condensation, our product was H2O. So one of our products was H2O. And we've also combined a molecule. So we've taken 2 smaller molecules and put them together in condensation, and we've made water in the process of doing so. So did we take 2 small molecules and make 1 big molecule? Not really. We didn't take 2 monomers or two parts and fuse them together. We just added water to the one other molecule that we had. So there was hydrolysis, was another reaction involving water. And in that case, H2O was one of the reactants, and it kind of is here. It's shown over the arrow here, so that's one of our small molecule reactants that might be shown over this arrow. And so this is kind of confusing with hydrolysis, except this. Hydrolysis has the word lysis in it. To lyse something means you're splitting it in half. Did we split this molecule in half? No, we didn't separate these carbons. We broke one bond, but we didn't break both. So if you take a look back at our hydrolysis examples, we've used that water to split the molecule. For hydration, we've just added the water to the molecule. The molecule overall remains intact. So that's the breakdown of the different types of reactions that we've studied so far that involve water. So this is our last slide for the Chapter 5 material. This one you don't necessarily need to know for the test, but I think it's kind of neat. The nature naturally selects which bond which of the carbons the oxygen is going to go to. So the carbon in the double bond that has more carbons bound to him is going to tend to be the one that gets oxygen more often. And the reason for that is a little bit complex, and we'd have to draw out the intermediate steps of this process. But that carbon is going to be a little more stable in terms of being able to pick up the OH part. Is going to be an intermediate, and it's able to hold this extra little charge that it has to hold because there's more carbons next to it that have more protons in their nucleus, so they can help share the burden there of the intermediate step. So this was first discovered by a Russian chemist, Vladimir Markovnikov, and this is called the Markovnikov rule. Again, you don't have to memorize that, but I think it's just a little neat organic chemistry tidbit. So that's it for chapter 5.